The E. coli cell membrane is composed of a phospholipid bilayer. Embedded proteins are integrated within the phospholipid bilayer of the cell membrane. Synthetic receptor proteins are designed to bind specific chemical inputs. Two distinct chemical inputs approach the cell membrane. Each chemical input binds to a specific site on a synthetic receptor protein. Upon binding of both inputs, the receptor protein undergoes a conformational change, initiating a signal cascade within the cell. The signal cascade initiates the activation of an internal circuit within the cell. This internal circuit is represented visually as a neon blue LED-style circuit. The circuit demonstrates AND gate logic, requiring both chemical inputs to activate and produce a visible output.
The cytoplasm is a dynamic environment within a bacterial cell, containing various cellular components. Plasmids are distinct circular DNA molecules found within the cytoplasm. Different colored plasmids indicate varied functions and states within the cell. A toggle switch mechanism involves two interlocking plasmid rings. The red plasmid expresses a repressor protein that inhibits the expression of the green plasmid, and vice versa. This mutually inhibitory relationship forms the basis of the toggle switch. An inducer molecule is introduced into the system. The inducer binds to the repressor protein, inactivating it and releasing inhibition. Release of inhibition allows expression of the previously repressed plasmid.
The bacterial cytosol is the internal fluid environment, filled with various molecules essential for cellular processes. Ribosomes, complex molecular machines, are distributed throughout the cytosol, facilitating protein synthesis. mRNA strands appear as distinct entities within the cytosol, carrying genetic information for protein production. Translation initiation begins with a ribosome attaching to an mRNA strand. The ribosome scans the mRNA for the start codon to initiate protein synthesis. Synthetic promoter circuits initiate transcription, which is followed by translation. Ribosomes synthesize fluorescent proteins as they move along the mRNA strand. Newly synthesized proteins emit light, indicating successful translation. Activation of the synthetic promoter circuit leads to reporter protein expression, driven by ribosome activity.
Saccharomyces cerevisiae, commonly known as baker's yeast, is a single-celled eukaryotic microorganism. The cytoplasm of a yeast cell contains various organelles, each with specific functions. The nucleus, a membrane-bound structure, houses the yeast cell's genetic material. Transcription factors bind to synthetic promoter arrays within the nucleus. These synthetic arrays are engineered to respond specifically to the transcription factors. The binding event is a precise molecular interaction, demonstrating the specificity of transcription factors. Transcription factors binding to promoter arrays initiates the expression of luminescent RNA. Luminescent RNA emits phase-shifted light pulses, dynamically displaying the logic gate's activity. Different light emission phases visualize the computational process within the cell.
Bacterial cytoplasm is a complex, crowded environment containing essential components for cellular function. Ribosomes within the cytoplasm are responsible for protein synthesis, translating genetic code into functional proteins. DNA, the genetic material, resides within the cytoplasm and directs cellular processes through gene expression. An enzyme cluster initiates a metabolic pathway within a cell. Substrate A enters the enzyme cluster and is transformed into metabolite B. Metabolite B moves towards the next enzyme cluster through a visually distinct pipeline. Metabolite B flows through a glowing pipeline towards an enzyme cluster. Within the enzyme cluster, metabolite B is converted into metabolite C. This sequential metabolic pathway powers bioproduction within the bacterial cell.
A bacterial cell secretes autoinducer molecules into its surrounding environment. Autoinducer molecules are small and diffuse freely. Brownian motion causes autoinducer molecules to move away from the cell in all directions. Autoinducer molecules diffuse through the intercellular space. Autoinducer molecules move towards a neighboring bacterial cell. The concentration of autoinducer molecules increases as more are secreted. Auto-inducer molecules bind to specific receptor proteins on the surface of neighboring bacterial cells. This binding activates a signaling pathway within the cell. Activation of the signaling pathway leads to the expression of specific genes, including a fluorescent reporter, causing the cell to fluoresce.
The yeast cell vacuole structure is clearly defined under high magnification. Initially, oscillator proteins are distributed evenly throughout the vacuole. This homogeneous distribution represents the initial state before oscillations begin. Synthetic oscillator proteins exhibit cyclical pulsing behavior within the yeast vacuole. These protein oscillations manifest as concentric waves emanating from a central point. The oscillation cycle demonstrates distinct peaks and troughs in protein concentration over time. Oscillator protein pulses drive rhythmic gene expression within the yeast vacuole. Alternating color bands within the vacuole visualize rhythmic gene expression. Protein oscillations directly correlate with and drive rhythmic changes in gene expression.
A T cell circulates within a simulated blood vessel environment. Initially, the synthetic toggle switch within the T cell is in the off state. In the off state, the T cell's plasma membrane shows no expression of CARs, chimeric antigen receptors. A synthetic toggle switch, upon receiving a specific signal, transitions to the on state. Activation of the toggle switch initiates the transcription of the CAR gene within the cell nucleus. The transcribed mRNA is then translated into the CAR protein in the cell's cytoplasm. With the toggle switch in the on position, CARs are expressed on the T cell surface. The plasma membrane visibly displays the expressed CARs. The T cell interacts with a target cancer cell, demonstrating CAR-mediated recognition and binding.
Intestinal villi are finger-like projections that increase the surface area of the small intestine. Epithelial cells line the surface of the villi, forming the gut lining. The gut lining provides the environment where the biofilm logic circuit operates. Engineered probiotic bacteria colonize the gut lining. Individual bacteria aggregate to iniziate biofilm formation. A structured community is created as the biofilm matures. Within the biofilm, individual cells flash in specific patterns, demonstrating the spatial logic circuit in action. Nutrient signals from the gut are processed by the bacteria, triggering different cellular responses. The biofilm functions as a logic gate, processing inputs and producing specific outputs based on cellular activity.
A yeast cell's interior contains various organelles, including mitochondria, which are essential for cellular metabolism. Mitochondria are distributed throughout the cytoplasm, facilitating energy production within the cell. The internal structure of mitochondria is optimized for efficient energy conversion through cellular respiration. Synthetic metabolic modules within the mitochondrial matrix facilitate various biochemical reactions. Each module is specialized for a specific metabolic pathway, ensuring parallel processing of multiple substrates. Fluctuations in module color intensity indicate real-time changes in substrate conversion rates. Metabolic pathways are conditionally activated by environmental cues such as nutrient availability and stress signals. Specific conditions trigger the activation or deactivation of metabolic modules. Increased pathway activity results in enhanced substrate processing within the activated metabolic module.